Hello and welcome to Stories of Science. Here, we look back on humanity's long journey to understand the world. The questions, the failures, and the breakthroughs that slowly changed how we see the universe and ourselves. We tell science as stories.
How Barbara McClintock revolutionized genetics, the jumping genes that changed science forever. Did you know that genes can jump? Yep, you heard that right. Barbara McClintock proposed a mind-blowing idea that left scientists in awe. Genes don't just sit still on chromosomes. They actually jump around. And no, this isn't from a sci-fi novel, it's McClintock's real discovery, jumping genes. If you thought genes were like books sitting neatly on a shelf, you're way off. Picture this, genes are no longer just hanging out in one spot. They sneak around, hopping to different parts of the chromosome, dancing like pros spinning, shifting positions, and totally breaking the rules. This discovery didn't just win her a Nobel Prize, it completely changed how we understand genes. Scientists had always believed genes were just the messengers of genetic information, passing it along according to strict rules. But McClintock came along and blew everyone's mind. Turns out, genes are a bit rebellious, they're not always well-behaved. Her research flipped the script on existing genetic theories and became a cornerstone of modern genetics. But getting here wasn't easy. At first, McClintock's discovery didn't get much respect. Many people thought she'd made a mistake or that it was just a fluke. But she didn't give up. She kept pushing forward, and eventually, she proved her theory right. In fact, it took decades before the scientific community fully accepted it. And when they did, the world recognized her brilliance with the Nobel Prize. So, next time you hear the term jumping genes, imagine those little genes as secret party dancers, gliding from one spot to another. That's the shocking impact McClintock brought to the world of science. Barbara McClintock's story begins in 1902, a year when the world was on the brink of major change. The Industrial Revolution was well underway, and the rapid development of science was opening new doors for humanity. Born in New York, McClintock came from a family that blended science and art. Her father was a doctor, and her mother was an artist. This unique mix shaped her mind, providing her with a balance of creativity and logic, which would become crucial for her future career. Imagine growing up in a home where deep discussions about medicine and the free spirit of art were part of daily life. This blend of influence fostered her broad way of thinking and encouraged her to challenge the status quo. In McClintock's home, knowledge and freedom went hand in hand. Her mother, the artist, valued creative thought, while her father, a scientist, gave her rigorous academic training. This dual influence allowed McClintock to think outside the box, merging the analytical with the creative. She learned how to view the world from different perspectives, question the norm, and venture into unknown territories. Her academic journey took her to Cornell University in New York, a place that would set the stage for her career. Unlike many of her peers who pursued fields like medicine or art, McClintock chose biology, a relatively unconventional path at the time. But Cornell opened her eyes to a world of academic possibilities, especially when she met T.H. Morgan, a giant in the field of genetics. Morgan's work with fruit flies had earned him a Nobel Prize and gave McClintock a deep understanding of genetic principles. This experience laid the foundation for her future scientific breakthroughs. However, McClintock wasn't content with simply inheriting existing knowledge. Deep down, she knew that science wasn't just about repeating what was already known, it was about exploring the unknown. Driven by curiosity, she challenged conventional thinking and pushed herself to find answers to unasked questions. In the late 1920s, McClintock began researching genes, choosing corn as her subject. At the time, corn wasn't a common model for genetic research, so many questioned her choice. But McClintock's persistence paid off. Her careful observation of genetic variations in corn led her to a groundbreaking discovery. By the early 1930s, her experiments began to show extraordinary results. She noticed that certain genes in corn weren't fixed in place as traditional genetics had suggested. Instead, they seemed to jump around on the chromosomes, challenging the very core of genetic theory. The idea of jumping genes was almost unbelievable to scientists back then, and many dismissed it as a mistake. But McClintock was convinced she was onto something. After years of research and experimentation, McClintock confirmed her hypothesis. She had discovered that some genes could indeed move freely between chromosomes, just like dancers performing a routine. She named these transposable elements, or jumping genes. Her discovery not only challenged the traditional belief that genes were stationary, but it also laid the groundwork for modern genetic engineering and molecular biology. Despite her groundbreaking work, McClintock's discovery wasn't immediately accepted by the scientific community. Her theory conflicted with the prevailing view of genetics, and many dismissed her findings as too radical. But McClintock didn't give up. She continued her research, fine-tuning her theory and providing the evidence needed to prove her findings were correct. By the 1960s, more and more scientists began to recognize the significance of McClintock's work, and by the 1970s, her theory became a cornerstone of genetics. In 1975, McClintock received the Nobel Prize in Physiology or Medicine for her pioneering contributions to genetics, becoming one of the few women to receive the Nobel for her work in the field. Her impact wasn't just confined to genetics. McClintock's discoveries helped spark rapid advances in molecular biology and genetic engineering. Her research provided the foundation for technologies like gene editing and gene therapy. The jumping gene theory transformed our understanding of genetics and opened new doors in the study of genetic variation. Though McClintock faced years of isolation and skepticism from the scientific community, she never wavered in her belief. Her story teaches us that science isn't just about following in others' footsteps, it's about challenging existing theories and seeking answers in uncharted territory. Her perseverance, courage, and relentless curiosity made her one of the most influential scientists in history. In the end, McClintock's life shows us that the road to discovery isn't always straightforward. Sometimes, it's about breaking the mold and daring to question the conventional wisdom. Her contributions continue to shape the future of science, and her legacy is a testament to the power of perseverance and bold thinking. As the saying goes, fortune favors the bold, and McClintock was undoubtedly bold in her pursuit of scientific truth. Barbara McClintock's groundbreaking discovery of jumping genes didn't just reshape our understanding of genetics, it challenged the very foundation of how we viewed genes. Back in the 1930s, when she first proposed her theory, the scientific world was skeptical at best. Genes, as the prevailing genetic models suggested, were supposed to be fixed in place, like little soldiers standing in line. But McClintock's findings, based on her careful study of corn, turned that idea upside down. At first, nobody believed her. She was dismissed, her work was doubted, and some even thought her results were just experimental errors. After all, corn wasn't the typical model for genetic research at the time. McClintock, however, wasn't the kind of scientist to back down from a challenge. She pressed on, refining her experiments and observations, convinced that she was onto something big. What she discovered was nothing short of revolutionary. Some genes, she found, could move from one chromosome to another, essentially jumping around. This wasn't just a random quirk, it was a fundamental shift in how genetics worked. Genes were no longer static, they had a dynamic, mobile nature that could alter the way traits were passed on. But even after documenting this jumping gene phenomenon, McClintock faced resistance. Critics argued that her findings were nothing more than an anomaly, a glitch in her experiments. She was ridiculed and largely ignored, but McClintock wasn't deterred. She knew her discovery was valid and kept refining her theory, even though it would take decades for the scientific community to fully accept it. Then, in the 1960s, molecular biology and new technologies like gene sequencing began to change everything. Scientists were now able to directly observe the movement of genes within the chromosome, and soon they found that McClintock's jumping genes were not only real, but crucial to understanding genetic variation. These mobile DNA segments, called transposable elements, were found in a wide range of organisms, not just corn. By the 1970s, McClintock's theory was embraced by the scientific community. Her work on transposable elements provided a crucial piece of the puzzle in understanding gene mutation, gene rearrangement, and even the evolution of antibiotic resistance. In fact, it's safe to say that without her discovery, modern genetics would have been missing a key component. As scientists began to explore the dynamic nature of genes, McClintock's theories were proven time and time again, paving the way for the future of gene editing, genetic therapies, and molecular biology. Thanks to McClintock, we now understand that genes are not just static carriers of information, they're constantly moving and shifting, like dancers at a ball, adapting and changing the genetic landscape of life itself. In fact, as the saying goes, if you don't go after what you want, you'll never get it. McClintock's dogged persistence in the face of doubt and ridicule led her to become a pioneer in genetics, and her legacy lives on in the very field she helped revolutionize. The world may not have listened at first, but today, her discovery is a cornerstone of modern genetics and molecular biology. So next time you hear about gene editing or gene therapy, remember McClintock's work. Without her, jumping genes might have never had their moment in the spotlight. She brought the dance floor to the cornfields and changed science forever. In 1983, Barbara McClintock's name was finally called out as a Nobel Prize winner. After decades of relentless work and dedication, it was a moment that felt like the perfect ending to her long journey. Imagine this: all those scientists who had once dismissed her theory of jumping genes must have been secretly regretting not recognizing her brilliance sooner. Why didn't we see it? They must have asked themselves. McClintock's Nobel wasn't just a win for her, it was a triumph of persistence and passion for science. Her research changed the way we understand genetics and opened the doors to molecular biology and genetic engineering. It was more than just a personal achievement, it was the ultimate acknowledgement of her groundbreaking work. But the road to the Nobel wasn't a straight path. Although McClintock's findings started gaining recognition in the 1960s, her theory didn't get the attention it deserved in her younger years. It wasn't until the 1970s, when the scientific community started warming up to her work on transposable elements, that her theories began to gain real traction. In 1983, at the age of 88, McClintock stood at the pinnacle of the scientific world, finally receiving the recognition she had earned long before. "It's a tremendous honor," she said, "but I'm most proud that my work has had such a profound impact on science." Those words summed up a lifetime of dedication. After years of being disregarded, McClintock had finally proven the world wrong. Her Nobel was not just a recognition of her personal achievements, it was a validation of the incredible value of her discovery. McClintock's work on transposable elements transformed our understanding of genetics. She showed that genes didn't just transfer information, they could actually jump between chromosomes, influencing an organism's genetic traits in ways no one had ever imagined before. Her findings set the stage for the next era of genetic research, opening the doors to genetic mutation, gene editing, and the groundbreaking technologies we use today. For McClintock, the Nobel wasn't just a medal, it was a symbol of her hard-fought journey. Think about it: her ideas were once labeled as wild fantasies, dismissed by the scientific community as unproven. But over time, as technology advanced and more research was done, it became clear her jumping gene theory wasn't just revolutionary, it was right. And those who doubted her? Well, it's safe to say some of them must have felt pretty embarrassed. Who would have thought that a female scientist, working in a cornfield, would uncover one of genetics' greatest secrets? As one scholar later put it, without McClintock, we'd never truly understand the concept of genes jumping. Her discovery didn't just give us a new way to think about genetics, it showed us that genes are not static, but dynamic. They move, change, and influence the world in ways we never thought possible. Like dancers on a stage, genes jump from one position to another, altering the genetic makeup of organisms. And through it all, McClintock's perseverance and courage were key. She didn't let the criticism or the skepticism derail her. Instead, she used it as fuel to keep pushing forward. "Science isn't just about discovering what's already known," she often said, "it's about breaking new ground." That mindset helped her make a contribution that changed the course of scientific history. In 1983, McClintock's name was finally inscribed in the list of Nobel laureates, but that achievement was not a coincidence. Her work didn't just introduce the concept of jumping genes to the world, it fundamentally changed the entire field of genetics. Before McClintock, scientists believed genes were fixed on chromosomes, unchanging. But her research proved that genes could move freely, like dancers leaping across a stage. This discovery not only changed how we view genetics but also paved the way for the rapid development of genetic engineering and gene editing technologies. What McClintock gave us was a new perspective. Genes weren't just carriers of heredity, they were dynamic and malleable. Her work opened a new era of genetic research, one that focuses on understanding how genes behave, function, and change. Today, thanks to her, we have a deeper understanding of gene mutations, gene repair, and genetic engineering. Her contributions continue to shape fields like medicine, agriculture, and biotechnology. As the saying goes, good things come to those who wait, and for McClintock, the Nobel was the culmination of a lifetime of perseverance. From being ridiculed to standing on the world stage, McClintock proved that science is not just about accepting the status quo, it's about challenging it, questioning it, and pushing the boundaries of what we know. Her jumping genes weren't just an academic footnote, they were a main event in the story of science. Barbara McClintock, a towering figure in the history of genetics, was known for her groundbreaking scientific discoveries. But when it came to her personal life, she remained relatively low-key. Unlike many other brilliant scientists, McClintock made a unique choice. She poured all her energy into her passion for research, leaving behind the conventional paths of marriage and family life. Perhaps, in a way, that's why her jumping genes were more inclined toward freedom. Her lifestyle and personality reflect her intense dedication to science. Though she had considered marriage and family in her younger years, McClintock ultimately chose to devote herself fully to her scientific work. Her closest friends and colleagues remember her for her deep love for science. McClintock's world was almost entirely consumed by the lab experiments, observations, and thoughts on the cutting edge of research. As she once said in an interview, "I don't feel the need for anything else to fill my life. Science gives me all the meaning I need." That pretty much sums up her philosophy. She had no desire to split her focus away from the world of science. It's not that McClintock lacked opportunities for marriage, she had a few intimate relationships throughout her life, but she never walked down the aisle. Her friends and colleagues all agree that McClintock was clear about her passion for science, and she cherished her independence and freedom. She didn't crave family life. In fact, she felt that marriage and family would take up too much of her time and energy, and she couldn't reconcile that with her devotion to her work. Her independence was one of the keys to her success. McClintock was fiercely self-aware, with a strong need for personal space. Her daily life was almost entirely centered around her research. From morning to night, you'd find her in the lab, immersed in her scientific world. Her colleagues and friends believed that this intense focus on her work was a major factor in her breakthroughs. She didn't need the distractions of social gatherings, her inner world was rich enough. As she once put it, "I don't need others to define my life. I can walk my own path." McClintock's independence extended beyond just her time management. She also didn't care much for the judgment of others. At a time when female scientists faced significant social pressures and biases, McClintock didn't let them affect her work. She focused on research, not on conforming to societal expectations. This level of determination and concentration may have been the very reason she was able to make groundbreaking discoveries in genetics. Interestingly, McClintock's theory of jumping genes seems to symbolize her approach to life in some ways. Just as genes don't stay fixed in one spot but jump freely between chromosomes, McClintock's life was dynamic, fluid, and untethered by traditional constraints. Perhaps she resonated with those jumping genes more than she realized. Her life, like the genes she studied, was about seeking freedom and independence. Her choice to live independently made her something of a scientific loner in the field. Her relationships with people, especially with others outside the realm of genetics, seemed far less intimate than her bond with genes. In a way, McClintock's jumping genes weren't just a scientific concept, they mirrored her own life. Her connection with genes was perhaps closer than her connection with other people. As she herself said, "My genes don't need to get married, they're already jumping freely between chromosomes." McClintock's quest for personal space and independence made her life stand out from the norm. She chose to dedicate herself fully to her research, committing all her time and energy to her scientific career and not being bound by society's traditional expectations. This choice allowed her to focus on her work and overcome many scientific challenges that others thought were insurmountable. In many ways, McClintock was a scientific single. Her relationship with her jumping genes seemed far more intimate than any human connection. Her personal life may not have been as famous as her scientific career, but her choice to remain single undoubtedly allowed her to live a life of freedom and focus. She is likely one of the most independent women in the history of science. She never let outside opinions sway her scientific direction, nor did she compromise to meet anyone's expectations. Every decision she made was to pursue her true love, science. Her lifestyle, while not conforming to traditional female expectations, perfectly aligned with her mission as an outstanding scientist. Her solitude wasn't a sign of boredom, it was in harmony with her pursuit. Her independence wasn't cold, it fueled her drive and creativity in science. McClintock's life was like the genes she studied, full of change, exploration, and a constant push toward freedom. Her life has certainly set an example for women scientists who dare to pursue their own path, standing strong in their beliefs. Ultimately, even though McClintock chose to remain single and focused on her research, her accomplishments and influence far surpassed her personal lifestyle. Her name will forever be etched in the history of genetics, offering a vital lesson to young scientists, especially women in science. Stay true to yourself, seek the truth, embrace solitude, and keep moving forward. Barbara McClintock's contributions didn't just make her a pioneer in genetics, they've had a lasting impact on multiple scientific fields. She's considered one of the founders of genetics, and her discovery of transposable elements laid the groundwork for modern gene editing technologies, while also providing fresh perspectives in cancer research. Today, when we talk about CRISPR, gene editing, and gene therapies, McClintock's work is quietly, but deeply, embedded in all of it. You might not even realize it, but every time you see news about gene editing, McClintock's shadow is likely there. She's not just a genetic pioneer, she's the grandmother of all gene editing technologies. If you've heard of CRISPR, you know it's revolutionized genetic research. CRISPR allows scientists to edit genes quickly and precisely, even offering hope for treating genetic disorders. But to understand how CRISPR came about, we need to look back at McClintock's discovery of transposable elements. Her research on the jumping genes in corn revealed the dynamic nature of genes, how they're not fixed, but rather capable of movement and change. This finding challenged the genetic theories of her time and introduced a completely new perspective: genes can interact, move, and evolve. McClintock's discovery of transposable elements gave rise to the theory that would later support the development of gene editing technologies. Scientists began to realize that genes aren't stationary, they're dynamic, and they can be rearranged, modified, or even deleted within the genome. While McClintock didn't directly contribute to the development of gene editing technologies, her work undoubtedly laid the theoretical foundation for them. For example, the rapid growth of gene editing tools like CRISPR is built upon her groundbreaking discovery of jumping genes. But McClintock's influence didn't stop with genetics. Her work also opened new avenues for cancer research. The role of transposable elements in cancer has been an important focus in genetic research. Many cancers, especially those linked to genetic mutations, involve genome instability and gene rearrangements. McClintock's research helped scientists realize that these jumps in the genome are not always harmless. In fact, they can sometimes trigger severe diseases like cancer. So next time you hear the phrase genes dancing, remember McClintock is the one who gave them the stage to perform. This isn't just a tribute to her groundbreaking contributions, it's also a nod to her relentless pursuit of truth. No matter when or where she lived, her legacy will continue to shape science. Just like her jumping genes, her name will forever shine on the stage of scientific discovery. In the end, McClintock's impact is more than just historical. Her discoveries have changed the course of modern science, making it possible to edit genes, understand disease mechanisms, and even fight cancer. Her work not only transformed genetics but continues to inspire scientists today. So let's give her a round of applause for setting the stage for the gene editing revolution.
The people who change the world usually just think one step deeper. If this story made you see things differently, hit like, subscribe, and save it. See you in the next story.